Scow, and this is soil microbiology, and I am finishing up now lecture 5, which is about metabolism, and we're in part 2 now, and this is going to be about flooded soils. And I have an interesting background. I'm in Washington, D.C., and the lighting is strange here, and there's this weird glow and changing of color that the camera is experiencing. So I'm going to be looking slightly psychedelic. It isn't that exciting, but bear with me. You don't want to be looking at me anyway. But I want to just go ahead and record this lecture. So flooded soils. So flooded soils is like the electron tower in nature, I like to call it. It's this wonderful case study demonstration of a lot of the concepts that we just covered, looking at the theory behind this essentially variety of different electron acceptors that microorganisms can use, and also the electron donors they can use, although flooded soils are more about the electron acceptors. And we're going to look a little bit at why we see this succession of different electron acceptors being used, being sort of dominating, either with depth or over time in the same place in flooded soils. And then we'll also look at other places where we see the same kind of phenomenon, but flooded soils are loud. And the other thing I want to remind you is that you have a lab on flooded soils where, by setting up these flasks that you added different things to, I wonder if you remember what you added to them. Think about them. So what you did is you set them up at different times, and then you're going to look at them all the same day, meaning that they are different ages, meaning they have had different amounts of times to kind of work through the different sorts of substrates that are available for microorganisms to use. And hopefully it'll be pretty nifty. It's a lab that usually works. So flooded soils are found in rice paddies, where we intentionally flood them, in wetlands, in compacted soils where it's not easy for soils to drain of water after an irrigation event or after rainfall. And immediately after rain or irrigation, there's this window of time when you can have these kinds of phenomena going on. And there's some beautiful scenes shown here of these different rice paddy systems, actually, in different parts of the world. This is not in California. So let's take a look at this figure to the left. And if you take a little bit of time and look at it, take a look at it and think about the electron tower, and now look at that flooded soil where we're looking at a cross section of soil. This is the top where there's atmosphere. Here's the water level. And here's about where the soil begins. So there's some water ponded on top. And then this is as we move deeper into the soil. And you can see that there's concentrations of different substances, and some of them are electron donors, and some of them are what? That's right, products of things being used as electron donors. And then to the left, there's these zones that correspond to different dominations by different kinds of electron acceptors being used, with oxygen aerobic being in the surface and methane being generated, which is actually CO2 being used as the electron acceptor, down in the bottom. So how does that compare to the electron tower? Does it seem like it's in the same order? What do you think? Yeah, it's flipped, isn't it? In the electron tower, we had oxygen at the bottom. Remember, the electron fell from the top, caught at oxygen, and then we had nitrate, then we had iron and manganese, then sulfate as we got higher, and then CO2. So what we're seeing, this phenomenon that we're seeing here, is in a soil that's reached some kind of stability. It's been flooded for a while. As you move down deeper into the soil profile, you're going to see a zone at the top where oxygen is used, but then that oxygen is going to be depleted, as you can see, within a very short distance. And then nitrate, which is present, it isn't being used for a certain depth, but then when the oxygen is all consumed, then the nitrate is used. Then we don't see the oxidized form of manganese here. We see the reduced form. Reduced form of manganese is the product of the oxidized form being used. So you can see here the appearance of this product of manganese being used as an electron acceptor, then appearance of iron being used as an electron acceptor. And then the sulfate, which has just been hanging out really nicely, nobody's using it in the surface layer. There's much better choices for things to use. But at some point then the sulfate's going to be utilized. And here's the product of it, the reduced form of sulfide created with the consumption of the sulfate. So sulfate being converted to hydrogen sulfide. And then when the sulfate is used up, then you see the methane start to form. And what's missing there? We're not seeing the CO2 disappear. So we can define these zones, the aerobic zone, then the nitrate reducing zone, which is nitrate being used as an electron acceptor, then manganese and iron reducing zones, then the sulfate reducing zone here, and methane generation, which is down deeper. It's the deepest area in the wetland. And in fact, those bubbles, which are methane being generated deep down in like a wetland, that's because that CO2 is not a very favorable electron acceptor. It's not going to be used until all the other ones are used. So it's going to be happening deep in the profile. And that rotten egg smell, this guy, hydrogen sulfide, that also is going to occur fairly deep in the soil. And you know that when if you've dug around in wetlands or beaches where there's estuaries, you don't hit that rotten egg kind of smell until you get down a little bit deeper. It's not necessarily right at the surface. So here's another great picture, which is showing the same thing that we saw over here, which is looking at concentrations of these different potential electron acceptors: oxygen, nitrate, manganese, that's +4, ferric iron +3, sulfate, and CO2. Over time, if these were all present, they're not always present in all ecosystems, but if they were all present, you would see the oxygen used first, then the nitrate, and then so on, sort of in this order of preference that corresponds roughly to how they fall in the electron tower. Oxygen is at the bottom, then nitrate and iron and manganese, then sulfate here, not what I'm showing you here, but the electron tower, and then CO2. Because for the same electron donor, right, you're going to get more energy if you use a electron acceptor that's going to be associated with a bigger delta E, a bigger gradient. For example, if glucose is the electron donor for all of these, the delta E is going to be biggest for oxygen, and then will decline in this order. So organisms that have oxygen are most competitive for that glucose, and so forth as we go along. And then down here, it's a beautiful little figure. It just shows you the products. Here are the metabolites of these reactions: oxygen, creates water. Nitrate is going to nitrite, or N2, depending on how far reduced it's going to be. Manganese 4 is going to 2+, iron 3+ goes to 2+, sulfate goes to H2S, and CO2 goes to methane. So the same succession of products over time. And remember, we can measure the parent compound or the product when we're looking at processes. So you can see there's a succession of decreasingly favorable electron acceptors for the same electron donor with depth, so over space, but also over time. You see the same succession in the same place, but this over time. What is the electron donor? We've been looking at electron acceptors. What's the electron donor in this flooded soil? It could be different things. But usually when you see this kind of phenomenon going on, it's the organic matter in the system, the soil organic matter or any plant residue that's lying around. Okay, so here are a number of half reactions for the processes we were just talking about. Aerobic respiration, denitrification, which is nitrate being used as an electron acceptor, manganese reduction and iron reduction being used as electron acceptors, then sulfate reduction, and then methane production. With fermentation, remember we talked about fermentation in between. So, and here's this EH, which you're going to measure in your flooded soil systems, which is a measure of the ability overall to accept or donate electrons. So if EH is very positive, that means there's lots of electrons in the system that can be donated if the right kinds of electron acceptors are there. But over time, as all these different substrates that can be used in anaerobic respiration are consumed, that EH is going to get less and less positive and start to become negative and become quite negative until we're down to minus 200 to 300 millivolts in for methane production. And you can see fermentation, which you remember from the lecture, spans a big swath of EHs. And you can see that here. So it's happening simultaneously to some of these other reactions. And we're going to see that that's important in a minute, as the fermentation is a very important processing of more complex organic compounds into simpler ones that some of the organisms that use these less favorable electron acceptors require for them to be able to carry out those reactions. So, and what happens in a flooded soil? Sorry for that. That's the hotel's heating system going on, and I hope you can still hear me. I'll just talk loud. But what happens is that in a flooded soil system, if the pH starts out lower, it's going to rise over time and stabilize at around pH 7. And it rises because a lot of protons are going to be consumed in these reactions, these reduction reactions, and the net result is a pH rise, and it poises at around 7 due to the carbonate in the system. Okay, so here's the same kind of thing being shown, but here I'm also giving you the delta G. So an estimated delta G for these different kinds of half reactions, and we don't need to spend a whole lot of time on it. Here are those equations again. Here's another measure of redox. So from a fairly high redox, which means there's a lot of potential for electrons to be donated, to a very negative redox down here. And look at the delta G for these different reactions as you move down with this really energetically favorable aerobic respiration and denitrification, and then it getting lousier and lousier and lousier. Again, this is with some model organic compound shown here as CH2O. As everything is sort of relative to that one particular electron donor. Of course, if there were other electron donors involved, you might have some different delta Gs. Okay, so this slightly complicated figure, but it's not too complicated, is it really? It is showing you the same kind of phenomenon that we were looking at in that first flooded soil picture, where we have a high, we have oxygen being used as an electron acceptor up here, and then we have this cascade, here we go, nitrate reduction, manganese and iron reduction, sulfate reduction, methane formation, or CO2 being used as an electron acceptor, producing methane. So same kind of thing we were looking at, but what's interesting is to look at what's going on with the carbon. So as we were talking about before, the electron donor, these organic matter that's around plant residue, and what happens to that plant residue in these flooded soils is it's going to be broken down into smaller pieces, and those big polymers are going to be broken down into monomers, and then they're going to ferment. Remember, we talked about fermentation, and we talked about like a mother bird, right? We talked about that more complex, even like glucose is not the simplest compound, is being essentially partially metabolized by the fermenters, who get a little bit of energy from using it, but then they spit out these marvelous waste products. We think they're marvelous as people. There are cheeses and sauerkraut and beer and kombucha and kefir and yogurt, and I can go on and on and on. But these are also really important for microorganisms to use in nature, and you can see that in particular these organisms that are further down in terms of how favorable their electron acceptors are, how good they are in being able to really make that delta large, and that delta E, they actually really rely on these breakdown products of fermenters. Like a lot of sulfate reducers cannot use glucose, and certainly methanogens cannot use glucose. So the fermenters are making simpler organic compounds, but they're also making acetate and hydrogen gas and CO2, which are the ingredients for making methane. Look at that, hydrogen and CO2, that's one pathway for formation of methane. So without the fermenters there, we would not have methanogenesis, or use of CO2 as an electron acceptor. So you have this food web and this complex set of interactions among different organisms that lead to this variety of different metabolism being supported. Okay, here's a couple of other interesting outcomes of this flooded soil that have to do with some of the chemistry that occurs once these transformations are made in the forms of the major elements. So we have now formation of Fe2+ reduced iron. Remember, the electron acceptor is ferric iron, is Fe3+, but now it's made ferrous iron, and that sulfate has been converted to hydrogen sulfide. And these two compounds, and actually a lot of other chemicals combined with sulfide to produce a variety of different kinds of sulfides. It's kind of obvious anyway, but ferrous sulfide is one such product. And the interesting thing about these indirect effects of microorganisms on the chemistry that then permits these other kinds of not biological but chemical transformations to occur lead to precipitation out of these new forms of elements. And for example, ferrous sulfide is one that's really created a lot. And so you might see for a while the ferrous sulfide, the Fe2 plus rising, and the H2S like increasing in concentration, and suddenly, boom, their concentrations get really low again. And you might think, oh, what happened? Did some oxygen come in and permit microorganisms to use them now as donors? But no, that's not the case. What happens is they combined chemically and formed a sulfide that is not very soluble, so it came out of solution and therefore could not be measured anymore. The other thing that does is this H2S, which is a gas, would otherwise rise to the surface, but it may get snagged and stay below in that sulfide. And the other interesting thing about those products is reduced iron and iron sulfide compounds have colors, and this peculiar color of reduced iron, this kind of blue-gray color, is called glaying. Glaying, that's what this is called. And what happens in soil, you often are going to have a mixture of zones that are indeed very reduced in these pockets, but anywhere where a little bit of oxygen gets in the system, that iron's going to oxidize very rapidly and turn red. So you can see like here, probably where the plant roots went down in the soil and brought some oxygen down, that iron, which is dominating this particular soil core, became oxidized and turns bright orange. So this is used as a indicator of how long a portion of the year the soil is actually wet, submerged, and anaerobic, which is a really important distinguishing feature of a kind of soil. Here's another soil profile showing you a more, a different kind of reduced form of iron with some other elements, and you can see this color of this sort of bleached out grayish color in this situation. So here's an indirect effect of microorganisms causing the iron sulfide, the ferrous sulfide, to fall out of solution, make this color, and then when any oxygen is introduced, it's going to be that reduced iron is going to be used as an electron donor. Oxygen will be used as the acceptor, where the root introduces oxygen, right? And then you're going to get this red color, and you get this mottling appearance. Okay, I told you that there's other places you're going to find sort of the electron tower, this flooded soil syndrome. A really interesting place that occurs is in contaminated areas, particularly when the contaminant is something like petroleum, which is really quite delicious, quite degradable. At least many components of petroleum are degradable. So here's an area where there's been like a barrel that was buried, and it's been there a long time. So this is a mature plume. It's had a long time to like leak into the groundwater, and groundwater, like these underground rivers, and so the direction of the flow of the groundwater is in this direction. So actually up here is the area of the groundwater. This is the vadose zone here, right? But it's the zone of the groundwater that has been polluted the longest, right? And then as you move further and further away and get up to the toe of the plume. That's like new area now, that groundwater plume, filled with petroleum, filled with electron donor, right? That is now entering into groundwater that hasn't had an electron donor, and there's still oxygen present. So at that toe, you're going to see use of oxygen as an electron acceptor. And as you move back into the older and older parts of the plume, remember, because the plume started here and then it just slowly moved, and it just got here, but it's been here longer, and here longer, and here longer. Okay, so you get that same kind of succession of electron acceptors being used, and you can find these zones where here you have a lot of dominated by nitrate reducers, here it's dominated by iron and manganese reducers, here it is by sulfate reducers, and in the oldest part of the plume, CO2 is being used as an electron acceptor, and methane is being created. By the way, don't get mixed up and think this has something to do with how close it is to the atmosphere. Right? Just assume that there's no oxygen gradient here, and there isn't in this case, or any oxygen that came in was used long ago. So don't think of oxygen coming in. This is just like the age of the plume. So that same kind of phenomena as the plume has been longer in the groundwater. And this is really important for remediation, because if you're going to go in and try to use naturally occurring microorganisms and try to give them what they need to help speed up this natural occurrence of degradation, you may consider what kind of microbial processes are going on in the particular area that you want to clean up, to think about what you need to add. Also, people use these indicators, like looking for evidence of, say, nitrate reduction or iron reduction, to prove to, like, regulators that, oh, it's okay, my pollutant is degrading. Look, I've got evidence that microbes are using it, because otherwise you'd have to argue that these electron acceptors wouldn't be used without there being this driving force of this pollutant. Here, regulator, look, give me 10 more years and I'll just keep doing this. I'll keep monitoring and make sure nothing goes wrong, but I don't need to do anything more to this contaminated site, because natural processes are taking care of it. And that's called natural attenuation. It's a very common way to clean up petroleum, is to just to provide that evidence. And it's because there's way too many areas to clean up these days. And this type of situation may not be considered as big a problem as something like a chlorinated solvent site. Okay, and then exciting is we see the same kind of phenomenon in a soil aggregate. And the bigger a soil aggregate is, the more you're going to see these different zones, right? So here we have an aggregate with big pores around it, and the oxygen coming in from the atmosphere all around the aggregate. So the outer layer of the aggregate is dominated by aerobic organisms that are using oxygen, it's the most favorable electron acceptor. But as you move in, the oxygen has all been consumed, so the organisms are going to turn to the next favorable electron acceptor, which is nitrate. I didn't put iron and manganese here, and maybe sulfate as you go deeper into the aggregate. A lot of what we see in aggregates is definitely these anaerobic sites where nitrate is being used as an electron acceptor. This is extremely important, because whenever you fertilize the soil, and you fertilize it with nitrate or you fertilize it with ammonium, which immediately turns to nitrate in agricultural soils, you can count on some portion of that nitrate moving into these soil aggregates, and you can count on that nitrate being converted to, I like this one the best here, that nitrate being converted to N2, if you're lucky, right? N2, which goes back to the atmosphere and is benign, but some of it may also convert to nitrous oxide, N2O, which is a very potent greenhouse gas. So we estimate that you could have 20 to 30% of the nitrate that you add being denitrified, being converted to nitrogen gas, and essentially just off-gassing from your soil, like you thought you were fertilizing your plant. But a lot of it's just going to go straight to the atmosphere as nitrogen gas, and a portion of that is going to actually not make it all the way to nitrogen gas, but form nitrous oxide and contribute to global warming. And what that percentage is, how much of that fertilizer goes to nitrous oxide and not all the way to N2, is an area of huge studies, especially for people who are trying to estimate the impact of different processes and different sectors on greenhouse gas production, and people who want to study how can we reduce emissions of greenhouse gases. It's a big area in California for agriculture. They are doing a lot of work on this because, and trying to figure out what percent of fertilizer is converted to nitrous oxide, because it is really a very significant impact of cropping systems, agricultural cropping systems, on greenhouse gas production. So we'll touch on that a little bit more in the nitrogen cycle, but you can count on there being these little mini electron towers in well aerated agricultural soil, plenty of oxygen. The plants are doing just fine. The oxygen is certainly around and between the aggregates, but it doesn't penetrate all the way in the aggregate. Therefore, inside the aggregate are places where oxygen has been consumed and does not move in, and then that turns on nitrate as the next electron acceptor, and possibly sulfate and further on. And there are these big significances of nitrate being used as an electron acceptor because of this formation of a greenhouse gas. Okay. I don't know why I'm showing you this. That there's a series of reactions that glucose going to CO2 go through, with oxygen being used as an electron acceptor. I'm not sure why this slide is here, and I can't remember, sorry. Okay, the other interesting thing is if you step back a little bit and look at this larger system, you can see that you've used up all these electron acceptors and you've produced all these potential electron donors. Particularly let's look at methane and H2S. And they're not going to go anywhere. They can't be used. I don't mean not going to go anywhere, but if they just stayed down there, they really couldn't be used as electron donors, because you need, that's right, electron acceptors for those reactions to occur, but all the electron acceptors have been reduced, so there really isn't much action there. But the interesting thing about H2S, if it doesn't form ferrous sulfide, right, and precipitate out, it is volatile, so it can move and will start to move out of the system. And same with methane. Methane for sure, and that's why we see ebullition, those bubbles that move up to the surface as gases, these big blobs of methane gas. They're going to rise to the surface. And indeed, in this oxic zone, where there is plenty of oxygen at the surface, right? These really good donors are now in an area where there is electron acceptor present, and they damn well are going to be utilized by organisms that will grow in biofilms like methanotrophs or sulfur oxidizers at the surface of the flooded system, maybe in films that form at the surface. And they're going to try to intercept as much of that methane or sulfide gas as they possibly can to use it as energy sources. Because remember, this would be a donor, and that would be a donor now. Okay, also you can see this kind of thing happening like in plants that pump oxygen down into the flooded soil, like below ground, like rice. in these erythemas. I can't remember what they're called, but some of these plants, not all plants, but some plants pump oxygen below ground. And if they do, then there would be a source of electron acceptor for these reduced forms of these elements that would be found down in those areas. So you might get some of that happening around plant roots. Cool, huh? And so organisms that are using these now, formerly known as electron acceptors, now electron donors, right? These different elements, those are lithotropes that are using those. Okay, and here's an interesting little case study about this. And this has happened a whole lot in the Netherlands in particular, but also you can see this in the bay and in any areas where there's estuaries. So the Dutch have very little land area, and really early on they learned to completely engineer their landscapes and essentially push back the sea to then open up what was sediment, estuaries, or or just seashore sediment for agriculture. And this required building these dams and having extensive pumping going on, because the water wants to come back in here, but they're just continually pumping water back out of these soils to keep them drained. And so you have this landmass that opens up that you can now farm. So engineers, extremely successful engineers, the Dutch, for a long time now. But what happens is that you have, especially around, along the seashore, along the estuarine areas, or just at the interface of the ocean and land, there's a lot of sulfur in those systems, a lot of sulfur in seawater. And so you have a lot of H2S that had been produced in these flooded muds, right? These, like, because that's what these soils used to be, were these muds lying underneath the shallow seawater in these estuaries. So there's tons of H2S there, and when you drain them, then all this oxygen enters into the system, and the byproduct of that is sulfuric acid, which is an extremely strong acid and dissociates very, very rapidly and strongly, producing tons of acidity. And that's what you see when these soils are first drained, is that they're impossibly acidic and no plant can grow in them. So that this process has to take its course for a while, and also a lot of lime is used to try to buffer some of this pH drop that happens. But after a while, this H2S will work itself out of the system, and with some good liming, your soils will have high enough pHs eventually that you can plant crops. But really interesting thing that's all about these donors and acceptors. Okay, so now the last piece of this lecture is anabolism. Remember, anabolism is the building up of cells, and everybody, all life forms, are made up of, generally the same set of elements, maybe slightly different proportions of them, but what we see in bacterial cells isn't a whole lot different from what we see in our cells. So the dominant element in cells, if you're looking at the dry weight, right, the dry weight proportion of it, because the biggest portion of the mass of a cell is going to be water, right? But in the dry weight, about 50% is carbon, which is the main backbone of most cellular material, and then oxygen at about 20%, then nitrogen at around 14%. And you can look here and read what these different elements are used for cell, and also what is the source? Where are these elements obtained from by different microorganisms? Like, what are these forms that are in the soil that they obtain them from? So nitrogen is next, then hydrogen, which is never limiting. It's always readily available. But the next one's going to be phosphorus, then sulfur, potassium, and then these other ones as well. And you go from macro to micronutrients, right, as you move down into these elements that are needed in much smaller amounts, except they are, of course, very, very important as well for cells to grow. And manganese, cobalt, zinc, copper, and molybdenum are an additional set of trace element cations that are also required by some microorganisms, and certain ones require these for their enzymes. For example, copper is used by some of the methanotrophs and is absolutely required for their enzymes, and maybe adding copper can increase their rates. And molybdenum is absolutely required by nitrogen fixers, by most nitrogen fixers, the symbiotic ones, and adding molybdenum or just having molybdenum present in the soil is absolutely essential for that process to occur, and without it, it won't. So the trace elements can be really important. Let's see. Okay, so all these different things are important, and you can see that carbon is usually just by the sheer mass of it in as a component of cells is often what is limiting microbial metabolism and growth in soil. And nitrogen is what you're usually going to see next, and then phosphorus, and so on down the line. Okay, so this is an overview of all the anabolic processes. So here's a cell, and you can see it's about 70% water, and the rest is these different compounds. And we talked before about the elements, but those elements are combined into this variety of different macromolecules, these polymeric compounds that make up the cell. There's a small portion that are not macromolecules, but they're small constituents. But then we have phospholipids, DNA, look at all our friends, RNA from the methods lecture, and then a whole lot of protein, right, and polysaccharides. So these are all these macromolecules, or these polymers, right? Macromolecules are polymers that make up the cell. So how do we relate these elements that we were looking at before that are needed by microbes and these complex macromolecules that all life forms have that are required for life as we know it? Well, this is a picture that from like here on in is inside the cell, and here's all these different processes that are going on that are taking these basic ingredients that are outside the cell, like these elements, but also simple organic compounds like sugars, amino acids, nucleic acids, fatty acids that are coming from plant residues breaking down or from dead organisms around you. It's just kinds of organic compounds floating outside, and they're going to be transported or diffuse into the cell. And what you're going to have first made up are these monomers, these monomeric compounds, these building blocks like thymine, adenine. Do these look familiar? TAGC, yeah, that go into making DNA, and then the fatty acids, which go into making lipids, and the amino acids, which go into making proteins, and some of these compounds that are made up of sugars that go into making, NAG and NAM go into making peptidoglycan. What isn't shown here are polysaccharides, which are made up from the sugars. So these monomers then are synthesized into polymers. So there's the DNA, which makes up chromosomes, RNA, ribosomes, lipids that go into membranes, and then proteins and peptides making up all these different enzymes, transport proteins, and then the peptidoglycan, right? So this is essentially the kind of roadmap of anabolism. And of course, we can turn all of these around, turn the arrows in the other direction, and we will see when, like, a plant residue enters the soil, it's going to be disassembled. All these complex molecules then are going to be broken back down into these constituents, into polymers and into monomers, and then mineralized, converted into the mineral compounds that make them up, that then allow plants, in particular, since they tend to process a lot of the material in terrestrial ecosystems, they will now become plant available and start the whole process over again. Now, nitrogen is this second most important element, not oxygen, but after carbon, then nitrogen is going to be most limiting. So nitrogen is taken up by glutamate dehydrogenase to make glutamate from alpha-ketoglutarate, and we're going to look to see where that is in a second. But that's is essentially just a couple of doorways or portals for nitrogen. There's that heater again to enter into the cell, into making amino acids that are the carriers, essentially, of nitrate to make all the other amino acids from. You may remember that from biochemistry, and we won't go into a lot of detail about that, but that's just something to point out about nitrogen. So we go back to the TCA cycle. Remember, we looked at it from the perspective of catabolism, from energy acquisition, and also all the other processes that hook up to it that we looked at, like glycolysis, and all these steps of glycolysis. Remember, to pyruvate, and then the preparatory reactions that make acetyl-CoA that go into the TCA cycle. Well, all these compounds involved, these intermediate compounds in this series of reactions that go from glucose as our poster child compound, but it could be other ones as well. A lot of them are key intermediate compounds, key intermediates that are the building blocks of all these different kinds of monomers that go into making the polymers of cells. So my point being that this pathway of glucose to pyruvate into the TCA cycle is not also involved in making energy, but it's also making building blocks for anabolism. And you can see here that I don't want to spend too much time with looking at all the different compounds and what are the families of monomers that are made from different key intermediates. But you can see, like, acetyl-CoA is really important for fatty acids that make lipids, right? That's the building block of that. Amino acids are made from a variety of different building block compounds that are both in the TCA cycle but also in this glycolysis region, and also the nucleic acids are being made from glucose being transformed into ribose, and through a set of complicated reactions making nucleic acids. And by the way, glucose also can go up here, and glucose is coming from carbohydrates, but it can also be energized and be used to make cell wall components and extracellular polymers. So my point being, these reactions that we looked at for catabolism are also really important for creating building blocks that are used for anabolism. Okay, so then, knowing that these central metabolic pathways that we're looking at here are really, really important, and they're highly conserved among organisms. Like, ours don't look that different from those of a bacterium. I mean, this is something that life has decided really works well for energy and building cellular material. So it's very conserved. So now I just want to look at how then does it relate to all of these variety of different substrates out into the environment, because we've had this sort of conservation of pathways, like this conservation, sort of a commitment to this, not like thousands and thousands of different kinds of pathways, although there's many more than what I'm showing you here, but the ones I'm showing you here are extremely important for lots of cells. A lot of cells use these pathways. So rather than there being this huge, huge diversity, this is kind of what's been committed to. So, but we have this huge diversity of different compounds out in the environment. So what happens to them, or how are they transformed that they ultimately then will flow into these central intermediate compounds and be used to get energy and build cells from it. And so what we see a lot going on out in the environment is that these complex substances, these big, a lot of times insoluble polymeric compounds that are, left over when a plant dies, or they're fats. They could be petroleum compounds like aliphatics or aromatics. They could be like some of the pesticides out there in the world that a lot of them have aromatic compounds in them, or they could be lignin that's really important in plants, especially shrubs and trees, right? So how do they interact with these central metabolic pathways? And without going into a lot of detail, you can look at, there's a whole variety of different reactions, which I call preparatory reactions, preparing them, modifying these into forms that then could shunt right into these central metabolic pathways. Therefore, things like waxes will be converted into lipids that then can go to fatty acids, go to acetyl-CoA, and go into the TCA cycle. Aliphatics can be used through beta oxidation, and that's going to create fatty acids. So even petroleum compounds, right? Not just plant residues or animals. It's like petroleum are going to use the same pathways, preparatory reactions that then result in fatty acids being produced. Aromatics are broken down by several different pathways. We'll talk a little bit about that later. Again, not in too much detail, but they are also funneled into a few general pathways for aromatics. Even though there's tremendous diversity of aromatics out in the environment, they are just going to be modified, like snip off that thing that's hanging off the ring, or modify the constituents so that then they can flow into these several aromatic pathways that are going to produce either pyruvate or acetyl-CoA or succinate, which then will also just flow right into the TCA cycle, or produce products that are upstream from it that will then flow down into the TCA cycle. And you can just see already how energy is going to be acquired and anabolism is going to occur. And then, as we talked before, ammonia, like alpha ketoglutarate, which is in the TCA cycle, is the portal, is the doorway for that nitrogen into the cell. And that will make glutamate, and that glutamate will carry that nitrogen around to all these different skeletons for making amino acids, because these are the carbon skeletons, but they still need the nitrogen, they need the amine group. And that glutamate, that is the former alpha ketoglutarate that now has this amine, is going to donate them these amine groups so they can truly become amino acids. Wow, how's that for a central metabolism on crack. And I know that a lot of this is lacking very key, crucial detail, but I'm just trying to give you a bigger picture of what's going on, and then you can go, if you're interested in understanding more about the metabolism, and fill in a lot of the blanks that I obviously left. Oh, I didn't tell you about the big polymers of, polysaccharides that make up plant residues, extracellular enzymes. We're going to break those things down, which are often repeating sugar units of different kinds, into sugars, and then those are going to go into it. So anyway, there's an overview, and hopefully it's not too painful, and you can understand the importance of these central metabolic pathways for organisms to make energy and build cells from this vast variety of different forms of organic compounds. We've been focusing on organotropes here, just nitrogen as a nutrient a little bit as well. But this vast variety of different opportunities out there, and how they interact with this very conserved small number of central metabolic pathways and central intermediate compounds. Okay, I think I just am giving you the enzymes again, reminding you that they're super important for these processes. And I also have a number of supplementary figures here. I'll go through them really quickly. This is just for your fun, for your own information, whatever you want to do with it. There's another way of showing an overview of anaerobic respiration. Just same thing that we were looking at before, but showing it slightly from a different angle. Like here, that nitrate can form all these different compounds, and showing the role of and relationship of fermentation in there. And there's another way to look at the electron tower. I think there was the suggestion that it might be nicer to put oxygen at the top, and then have the as the best oxidants, the most favorable electron acceptors, and then you can move down below and get to the lower ones. And this shows it that way. So for those of you who like to see it the other way around, here is a relatively complex, but tells it like it is, depiction, conceptual model for the same kind of thing, but letting you look at in a different way. But also you can use this to predict which combinations of half reactions. Remember, it takes two redox pairs to tango, so this gives you a better idea of which ones you can actually couple with one another. Okay, and this is the in gory detail about the amino acid families that we just touched upon in the TCA cycle and glycolysis. Remember, we showed all these different building blocks for amino acids. Here are these families of amino acids that are made from these different building blocks that are shown over on the left-hand side. And a little bit about lipids and sugars there, including the importance of using ATP to turn glucose into a high energy form that can be used to make polysaccharides. So you have to pay to turn that glucose if you have a high carbon to nitrogen meal given to you and you can't build cells, right, because you have too much carbon and not enough nitrogen, but you're still greedy and you want to make polysaccharides and maybe excrete them and build aggregates, right, from the extracellular stuff that you release. Well, you need to pay for that. You need to add ATP to energize that glucose to be able to make polysaccharides. And autotrophs, how do they get carbon? So this is just reminding you that the Calvin cycle is there making the carbon compounds for autotrophs, because again, we were really focused on heterotrophs in this lecture, and particularly in looking at anabolism. And it costs, right? It costs to make organic compounds using the Calvin cycle. You need to use ATP and NADPH. So those poor autotrophs pay for their carbon. It isn't automatic. They don't just pull it out of the atmosphere. They pay to convert that CO2 and reduce it and convert it into sugars. And here is a conceptual model of a cell that shows it's a depiction of this bioinformatic information from a genome sequencing project, sequencing the whole genome of a particular prokaryotic organism, and then taking all that different information of all these different genes and showing their interconnections in this figure, just showing how really complicated and interrelated are these different types of metabolic reactions going on, that if you look hard you can kind of see the TCA cycle and some of our old friends in here, but there's also other things going on. Oh yes, and here's another one showing an organization of all the genes that are shown here around the outside, including for transport proteins that carry things into the cell, and then for the pathways inside the cell that carry out all the reactions that we were looking at. And look at, just so you feel comfortable, there's glycolysis going to pyruvate and the TCA cycle, and then you can see exactly the same things that we were looking at, but this is actually a model of the whole genome of a Pseudomonas, where they sequenced all the genes for all those different processes we were just looking at. And that's just another picture for you for fun. And that's the end. Okay. Thanks a lot.